curd and cheese microorganisms are needed. A bacterium called lactobacillus converts the lactose sugar present in a milk to lactic, which result in a conservation of milk into curd, which is really very much well known for, I think, almost all our attendees. So we can understand quite loudly that the science of microbiology aim to gain and expand our fundamental understanding of microorganisms by studying their microbiology, metabolism, physiology, reproduction, and genetics. This is all how microbiology plays a significant role in different sectors. In the next couple of years, or maybe say after 5 years or 10 years, we will see various other uses of microbiology which will be beneficial for us in all aspects of our life. So dear colleague, so our speaker of this session is none other than our very own faculty, Dr. Shashank Shivaji Kamble. He is an assistant professor in the Department of Biological Science at the School of Basic and Applied Sciences at G.D. Goenka University. Dr. Kamble has expertise in the area of biology of infectious disease with a background in microbiology. Dr. Kamble has served as the National Postdoctoral Fellow DST under DST project and as a research scientist at Jawaharlal Nehru University, New Delhi. Dr. Kamble earned a Ph.D. with a specialization in tuberculosis from CSIR Institute of Genomics and Integrative Biology. Dr. Kamble has more than 8 years of research and about more than 2 years of teaching experience. So as I just focused a bit about this session, so it is actually the microbiology, as I said, microbiology is the science of tiny organisms that are not visible to the naked eyes. Despite being so tiny, they can cause a life-threatening infection and pose a serious threat to the existence of mankind. So the recent pandemic of COVID-19 is one of the biggest examples of this. So on the other hand, these microorganisms also have a beneficial role such as fermentation, brewing, and production of useful metabolic products such as antibiotic, as I already mentioned earlier. So in this session, a student will get introduced to the role of microorganisms behind the day-to-day household activities such as cooking and self-hygiene. It will also highlight the importance of the normal microflora of a body in preventing hazardous infectious disease. This will create interest in them about tiny microorganisms and desire to study them. So, dear colleagues, in just a short information, I'm sure you understood how important it is, and therefore I may kindly request all of you to listen carefully the session. At the end of the session, we have let's say team of dedicated faculty members. They will collect your question. I may request you to drop your question on the chat box. Our faculty member will collect your question, and at the end of the session, we'll ask either you or our faculty member will ask a question on your behalf. And resource person, along with we have also some other relevant faculty members here besides Dr. Kamble. So all together we will try to condense your question. So before inviting the speaker, I may kindly request all of you to mute your mic and also mute your camera so that a speaker can conduct the session very smoothly. Well, so thank you all the attendees. I'm sure all this session will provoke your deep thinking. And now I may invite Dr. Kamble, Dr. Shashank Shivaji Kamble, to initiate your talk and discussion. So, Dr. Kamble.
Thank you, sir. Thank you. Thanks a lot.
Yeah, please.
So, thank you, sir, for the nice introduction. I hope I am audible to everyone.
Yes.
Yes, sir.
And I hope everyone can see my slides.
Yes, we can see your slide.
Okay. So, today, thank you, sir. So, today is the fifth session in the online student development program, which is organized by the School of Basic and Applied Sciences, GD Goenka University, Gurgaon. So, today I'll be talking about the microbial. We know about microbiology is the study of those organisms which are not visible to naked eyes. And the recent, as the deans has said, the recent COVID-19 pandemic is an example of that, and how such a tiny microorganism can cause a life-threatening disease to so many human beings. Okay, so we know that microbiology is the study of these organisms from the infectious point of view, but these microorganisms also have a very important role to play in the physiology of your body, and also the development of many of the organs, also the development of the immune system of your body, etc., etc. And also these microorganisms are very much essential in from the point of view of many industries such as food industry, brewing industry, fermentation industry, etc., etc. So we would look at the role of the microorganisms apart from the infectious diseases. So let's talk about microbial diversity. Microbial diversity, if you talk about no one can measure the microorganisms, the total number of microorganisms in the world, because the techniques that we have to measure those the numbers is very limited. We have huge limitations with our abilities, because the microorganisms, they can exist at all the geographical locations where a human being cannot reach. Okay, and this can include the North Pole, South Pole, to very high temperature volcanic eruptions, etc., etc., and even at the subsurface of marine and continents. So these are some of the locations where a human being cannot reach. So you can understand how difficult it is to estimate how many of the total microbial number exist on the planet Earth. If I go further, these are the methods that we currently use to assess the diversity of the microorganisms, both the functional diversity and the microbial diversity. When I say microbial diversity refers to the number of the microorganisms in a particular geographical area, whereas the functional diversity refers to the role of that microorganism in that particular community or in the ecosystem. So there are two different methods to measure this diversity: culture dependent method, culture independent method. In culture dependent method, what we do is that we try to mimic the natural ecosystem in the laboratory, and we try to give the microorganism the same conditions that they encounter in their natural ecosystem. Okay, and by doing that, we try to grow them first, and after growing them, then we try to study their growth pattern, their growth characteristics, their nutritional requirements, their biochemical characteristics, what is their is made up of, okay, and what is the genome is made up of, whether it's a DNA genome, whether it's an RNA genome. And in case of culture independent method, you go for genome sequencing, where you study what kind of genes are present in that particular microorganism, what kind of genes are getting expressed, and what kind of proteins are being made under a particular condition. Now, I think all of you are aware about the central dogma of biology, that is the genes. The DNA gives rise to RNA, and from the RNA, what we get is a functional protein. So proteins is the functional entity of DNA. Okay, and therefore, connecting the genomic constitution of a microorganism to its protein constitution is very much important to understand the various missing links. Okay, so one person said, this is a very big person, very big biologist. He said that we are inhabited by as many as 10,000 bacterial species, not number bacterial species. Those cells outnumber those which we consider as our own by the ratio of 10:1. And weight. So if you look at the total weight of the microorganism in your body, it's about 3 pounds, and it is the same weight as your brain also. So together, all these microorganisms of our body are referred to as the microbiome, and they play a very crucial role in our lives. so that in the today's life, scientists have begun to rediscover or reconsider what is the role of microorganisms apart from the infections that they cause. Is there any role of microorganisms in the development of the human being? And the answer is yes. So the Human Microbiome Project was undertaken by scientists so many years ago, and they have sequenced so many of the microorganisms from the human body, and now we know what is the normal microflora of the skin, of the lungs, of the gastrointestinal tract, of the GI tract, etc., etc. I hope I'm audible to everyone. Please let me know if I'm not audible.
Yeah, you are audible.
Yeah. So now let's look at what is the importance of this normal microflora, or we also call as microbiota. So if we look at this diagram, the microorganisms, the number of the microorganisms in our body is around 100 trillions. That is 10 times more than the number of our own cells. Okay, and if you compare the total number of genes that are present in this bacteria, in these microorganisms, is around 3 million genes. Okay, whereas if you compare the number of total genes in the human cells, then it is around 23,000 genes. So now you see in the number of genes also there is a difference of tenfold. And now we can see the microorganisms in our body, they easily outnumber the our very own cells. Okay, now let's look at what is the interaction between the human cells or the human body and the microorganisms which they possess. The microorganisms, they basically take the nutrition from the human body. However, the human body also try to kill them by using its immune system. And so there is a one beneficial part, there is one detrimental part. If you talk from the microorganism point of view, the microorganisms produce so many beneficial metabolites, especially in the gut. There are many vitamins which are produced in your gut by the microorganisms, and which you normally won't get from the food that you eat. Okay, for that you have to follow strict diet regime. So this bacteria actually helps you to produce so many vitamins and essential metabolites. Whereas on the other hand, they also produce certain toxins which can be detrimental for your body. So there is one beneficial effect and one harmful effect from the microbes point of view, and also from the host point of view. If you see on the right hand side, there is a bar. So which says that this seesaw that you will see, that you have seen over here, is a transient phase. Okay, so either it will lead to a disease condition where the toxins of the microorganism they prevail, or it will lead to a healthy relationship between the microorganisms and the human body. Okay, and both of these microorganisms and the human body, both of them are influenced by the environment that they live in. So the term normal microbiota is actually refers to the population of the microorganisms that inhabits the skin and the mucous membranes of the healthy normal person. This flora can be of two types. One is the resident flora, which is a permanent flora over there in a body part, and another one is a transient flora, which comes and goes. So the resident flora actually consists of relatively few number or the fixed number of microorganisms which are regularly found in a given area at a given age. If disturbed, so if this flora gets disturbed, then it can re-establish also itself. Okay, given a certain period of time for it to recover from the attack. So the transient flora is the one which generally consists of non-pathogenic or can be potentially pathogenic microorganisms that inhabit your body for, let's say, hours, days, or weeks. So it consists of so these organisms, transient flora, can colonize your body only when the normal or the resident flora is not there. So when you wash your hands regularly, very commonly with soaps, it generally tends to reduce your resident microbial flora, and that allows the transient flora to get established and form colonies. Okay, so therefore it is even the COVID-19 times, it is said that you should wash your hands at every now and then, but you should limit that thing to certain, let's say 5 times, 6 times a day, because more common washing of hands with soap might disturb your resident microflora of the skin, that might lead to other skin infections. Okay, so the transient flora is derived from the environment. It does not produce disease and does not establish itself permanently on the surface. Therefore, it is generally of very little significance. Okay, because when the normal flora, resident flora, it remains intact, then this transient flora has a very little significance. Okay. However, if the resident flora gets disturbed, perturbed, etc., due to whatever conditions, let's say physical injury, chemical injury, whatever it is, then these transient organisms, they can colonize, they can proliferate, they can form their own community, and then they can produce a disease. So now, there was a very important study. So the human microbiome project actually came to a conclusion that when they took the samples from 27 distinct body sites, which are rich in moisture and nutrients for the microorganisms, they found out that that out of 22 bacterial phyla which were observed in this 27 sites, around 92% of the genomic sequences belong to only 4 phyla. That means this 4 phylum of bacteria actually predominates your body. Okay, and the rest 8% belongs to the other bacteria. Okay, so now you can understand the importance of these 4 phyla. And which are these 4 phyla? One is, of course, the actinobacteria, then there is a firmicutes, proteobacteria, and bacteroids. Actinobacteria, generally the species or the strains which are found out in your body from Actinobacteria group are Corynebacterium, Propionibacterium, Microbacterium, Micrococcus. From Firmicutes group, generally the Staphylococcus aureus, Staphylococcus aureus, the Clostridium, they are generally observed. From the bacteroids, Sphingobacterium and Chrysobacterium are observed. And from the Proteobacteria, Pseudomonas, Xanthinobacterium, Halomonas, Dielsalfia, etc. are observed. Now, there was one more ambitious study which basically tend to discover the age-wise modulation of these 4 phyla, and it was observed not only single. There's not a single phylum which actually predominates a particular age group or a particular physiological condition, but it is the proportion. It's the proportion of. Hello.
Sir, you continue.
Okay. So it was observed that not a single bacteria actually affects the proportion of the bacteria. Not a single phyla actually affects the proportion of the microbiome in your body part according to age or according to physiological condition, but it is the proportion of all of these 4 phyla. So you can very easily see that that in case of the newborn, the actinobacteria are prevalent because the actinobacteria are the ones which produce essentially metabolites in your gut and other body parts, which actually are antimicrobial in origin. Antimicrobial in their properties. So actinobacteria produce a lot of other metabolites which kills other pathogenic organisms. So therefore, the newborn babies, the breastfed babies, they are inhibited by this actinobacteria to a very large level. And also one more fact, this actinobacteria are also present on the skin surfaces. So whenever a baby goes for breastfeeding, obviously the actinobacteria which are what you can say, which are the habitants of the breast skin, they get inside the mouth of the baby, and that is how they go to the gut of the baby. If you talk about the antibiotic treatment, in case of a person who is undergoing antibiotic treatment, bacteria generally prevail because bacteroids generally consist of a group of bacteria which can very easily fluctuate and modulate or even adapt to new antibiotics. So bacteroids are very easily adaptive to the antibiotic conditions. Therefore, they prevail in case of antibiotic treatment. If you talk about malnutrition, in case of malnutrition, proteobacteria prefer. So proteobacteria are the bacteria which converts the lactic acid that is produced in your body from the metabolism of glucose and other carbon sources into butyric acid. So butyric acid is basically a sign of putrefaction, that is fermentation. So malnourished people, they show signs of fermentation in their body, and therefore their body is not a aerobic chamber anymore. They more commonly produces anaerobic gases. And if you talk about the obese individuals, in case of obese, the firmicutes, they prevail along with the proteobacteria. Now, if you compare the healthy and obese, here also the firmicutes are more, but the level of proteobacteria is very less. So you might think that these two are very similar, almost same, but the proportion of proteobacteria is very different. Okay, and if you compare the people of more than 65 years of age, then the firmicutes prevail because firmicutes actually are the soil bacteria. Okay, so firmicutes also are a type of scavenging bacteria. So they also live on dead and decaying material. So firmicutes also exhibit what is called as a putrefaction, scavenging activity, and therefore, when the immune system goes down in your old age, the putrefaction starts, and that is how the firmicutes they grow. The host and the microflora interaction can be of multiple types. It can be of commensalism origin, like in commensalism, one organism benefits whereas the other one is unaffected. Whereas it can be a mutualism also, where both the organisms benefit from the interaction, or it can be parasitic interaction where one organism benefits at the expense of the other. Okay, so these microorganisms which are present on your body can be commensals, they can be facultative pathogens. What are facultative pathogens? That means they're opportunistic pathogen. If they're given a chance, then they will cause an infection, or they can be true pathogen also. And saprophytes actually refers to the scavenging microorganisms, which basically feeds on dead and decaying material. So there are some organs in your body which are sterile. Okay, so internal organs except the alimentary tract, that is your GI tract, they are sterile in healthy individuals. This is very important in healthy individuals. So the sterility is maintained by the local defense mechanisms, the physical barriers, the physiological barriers, the chemical barriers, as well as the immune cells. So if I were to give an example of physical barriers, skin is the best example. Skin is very tight. It has three different layers, and it's very difficult for the microorganisms to penetrate that thing. Until unless there is a wound, the microorganism find very difficult to penetrate your skin. If you talk about the urine has acidic pH. It also has a bile salts. So this physiological and physical factors, barriers, they basically prevent the growth of microorganisms in the sites. So the area of the body with the normal microflora is basically respiratory tract, the GI tract, the urogenital tract, and the skin. So what happens when? So what is the normal microflora of a newborn baby? So all of you should remember that inside the womb of a mother, the baby is generally in a sterile condition until unless the mother is infected by a virus which crosses the placental barrier and enters the body of the fetus. Until unless that point, the baby is almost devoid of all the microorganisms. Okay. However, as soon as it gets delivered, it is exposed to so many different microorganisms from the mother's body as well as from the outside environment, and this is how it develops its own microflora. So its oronasopharyngeal cavity, which is basically the respiratory and the oral route. That microflora develops due to dietary sources, the kind of food that the baby eat. It can be developed due to the breast milk. Okay, it can be also developed due to the environmental sources. In case of skin microflora, obviously because the baby gets delivered to the vaginal microorganisms, they play a very essential role in developing the microflora of the skin of the baby. And also, obviously, environmental sources play a very important role. Now, if you look at very carefully, because the baby is born, it is outside of the mother's womb, the environmental sources play a very, very important role in developing the normal microflora of all of his body parts. Okay. If you talk about the urogenital tract, it's again the vaginal birth canal, that is the vagina, which the microflora of the vagina plays a very essential role, and so on and so forth. And see, all of this basically depends on the basically what you can say, the condition of the mother also, like the age of the mother, because the vaginal microflora of the mother will change with the age. Also, whether she's malnourished or no, what is the gestation period, how much time was the baby inside the womb, whether it's a premature baby or it's normal baby. It will also so the microflora of the newborn baby will also depend on all of these factors, which are greatly influenced. So, in case of skin, the normal microflora, skin is the largest organ. It is a very large surface area, and basically, it is a kind of unfavorable habitat for the microorganisms. Why? Because skin, as you all know, we constantly produce sweat. The sweat contains sodium chloride, and therefore, the sodium chloride has a high concentration on the skin. And the sodium chloride is known to perturb the osmotic pressure of the bacterial cells and cause cell death. Also, the skin is a relatively dry surface as compared to other areas. And also the sebaceous glands that you can see in the diagram, they produce many fatty acids. Okay, and these fatty acids, they create localized acidic pH. Okay, and there are some bacteria which basically uses this fatty acids and convert it into lactic acid also. So all these acids basically contribute to the acidic pH of the skin, and that is why the skin is a relatively unfavorable habitat for the microorganisms. The normal microflora of the skin normally consists of the Staphylococcus epidermidis, which normally makes up more than 90% of this skin microflora. Then Staph aureus, which is prevalent in nose, perineum, and vulvar skin, and the Propionibacterium acne, which causes acne. Okay, pimples. So Propionibacterium acne generally is not found in the children below 10 years of age, but it is generally found in case of adolescent individuals. What happens, what are the causes for this altered skin microbiota? So the skin microbiota can be altered due to your, first of all, hygiene habits of hygiene, like regular hand washing, then having using local antibiotics, the ointment that you use, right? Soframycin, Bernol, etc., etc. Repeated use of that can even change the skin microflora. Along with that, the kind of diet that you use also has a very important effect on the skin microflora. The community that you live in, for example, you are living along with pets like dogs, cats, etc., etc. So the microflora from their skin can get transferred to your own skin also, and if given a chance, it can multiply, it can colonize your skin also, and can stay over there. Okay, stress also has a very important role to play in alteration of the skin microflora, and along with that, the age and the lifestyle. Lifestyle is very important, because the more physically active you are, the more outdoor games you play, the healthy microflora you will have. And if you're just sitting outside, inside, and eating and playing indoor games, then your microflora will not be healthy one. Okay, so all these basically factors play a very important role in altered skin microflora, and this altered skin microflora then will have a negative impact on the microbe host interaction and the microbe interaction, and this altered interaction can lead to chronic disease. If this persists for a long period of time, then it can cause a chronic disease. The normal microflora of the respiratory tract. So respiratory tract generally consists of the upper respiratory tract and lower respiratory tract. The lower respiratory tract consists of trachea and lungs, which are generally devoid of microorganisms. And the most of the microorganisms are generally found in the upper respiratory tract, which basically consists of nasopharynx, oral cavity, and throat. Okay. In case of oral cavity, the normal microflora consists at birth. So generally at birth, the mouth is generally sterile, but after the birth, within 4 to 12 hours, generally the baby acquire the bacteria, let's say from the milk, breast milk, or from the vaginal microflora, etc., etc., or from the environment also. Okay, when the neonates, when the baby grows to somewhat size, then the Streptococcus salivarius, which basically inhabits saliva. Other Staphylococcus, Neisseria, and other bacteria, they prevail. When the tooth appears, Streptococcus mutans generally prevail, and this bacteria is responsible for tooth decay. Whenever when you're in childhood, you must have got the tooth decay, gum infection. That is because of this bacteria, Streptococcus mutans. At puberty, the bacterioids and spirochetes, they are more commonly found in the mouth, and that is why we more commonly have a bad breath in the adult stage than in the child stage, because bacterioids and spirochetes, they produce various metabolites which have foul odor. So if you think of 1 ml of saliva, there are 10 to 8 bacteria per ml of saliva, and that corresponds to more than 700 different bacterial species or microbial species. So if you talk about the most, the area of your body which is mostly inhabited by microorganisms is basically gut. So gut, if you talk about the GI tract, it consists of stomach, it consists of the small intestine and large intestine. So in the stomach, the partial digestion of food happens, whereas in small intestine, the carbohydrates, the proteins, they get digested, and it's the large intestine in which the reabsorption of the nutrients into the bloodstream occurs. Okay, so stomach, because it has a very low pH and it also has a high concentration of bile salts, generally it contains very less bacteria. So most commonly, the bacteria which can inhabit the stomach are lactobacillus and the streptococci. In case of of small intestine, Lactobacilli, Escherichia coli, and Enterococcus faecalis they prevail. And Enterococcus faecalis is a very common very commonly found organism in your feces sample also. If you talk about the colon is generally consisting of anaerobic gases. Okay, so because in the colon, generally what happens is that whenever the undigested food it goes into the colon has a very rich microbial flora which digests these undigested carbohydrates and other molecules, and it makes anaerobic gases, and it also makes its own food for its living. And that therefore this colon has a relatively anaerobic microbial flora. This gut microflora can be changed with the age, with the geographical location, with the dietary habits, with the malnutrition, and very importantly with the use of antibiotics. And therefore it is recommended not to use antibiotics every now and then. If you try to use antibiotics every now and then, you're disturbing your own gut microflora, and you are allowing the pathogenic organisms to colonize your gut microflora, which ultimately can cause disease and even can cause you life-threatening conditions. So this is how the gut homeostasis is perturbed. So you can see on the left hand side, the one you can see in green is basically the normal microflora of your gut, and it has a very important role to play in your gut immune system, in the way you behave, because the metabolites which are produced by this gut microflora actually stimulates good feelings in your brain. They can cross the blood-brain barrier, and they can reach the CNS, central nervous system, where they can affect the good mood of good mood in your body, in your brain, sorry. And if you talk about the perturbation of this homeostasis, let's say there are many factors which play a role in this, like the reduced peristalsis, like reduced movement of the gut, which basically is responsible for the good bowel movement. So if you have a constipation, then you can have problem. If you have an immune suppression, which is generally normally when you take steroids, the people who goes to gym and who tries to make their body bigger in size, they generally use steroids. So steroids, they tend to produce immune suppression, and if you take it for a long period of time, it can cause you a huge loss. Okay, in case of physical destruction, like in case of ingestion of a chemical or burns, also this kind of hemostasis is perturbed. And very importantly, antimicrobial drugs, if you take in huge quantity for a long period of time, they can even perturb your gut homeostasis. And this generally leads to the initial symptom is the dysentery, diarrhea for a long period of time, and then you start to develop bad mood, bad behavior, and slowly it can lead to chronic conditions. The gut brain axis, as I told you before also, the microflora of your gut, they have a very important role to play in the way you behave. Okay, is it because this microflora, they basically activate the neural afferent circuits to the brain. They also induce the immune response of the mucosa, and they also produce metabolites which directly influence the CNS system. And therefore it is always said that keep your gut clean or good to keep your brain good. Okay, if your gut is bad, your brain will be bad, and therefore your behavior will be bad. Okay. So when we have a bad stomach, all of us must be knowing that we take probiotics. Okay, so we only know probiotics. There are 4 biotics which are very essential and which are generally prescribed to the patients which have a which have a chronic gut problem, which have chronic constipation, chronic diarrhea, chronic dysentery. So in all those patients in which there is a perturbed gut homeostasis for a long period of time, generally they are prescribed with these all biotics, okay, by the doctors. And this 4 types of biotics are probiotics, prebiotics, symbiotics, and metabiotics. Probiotics are basically the live bacteria. So you eat the live bacteria, okay, and those live bacteria are actually a part of your intestine only. So you eat them from outside. That is only the case. Okay, and basically why you do that? Because you want to increase their numbers. Okay, and these are the good bacteria. The symbiotics generally the prebiotics consist of adding the food. In case of prebiotics, you don't eat the bacteria, you eat the food for the bacteria. Like as I told you, in case of colon, in the colon, the undigested carbohydrates or proteins, they accumulate. Okay, and this is the food for the gut microflora. Okay, so in case of prebiotics, you eat their food. You eat the carbohydrates which are not digestible by your own body. So obviously they will accumulate in your intestine, in the colon, and obviously they will be metabolized by the bacteria. In case of symbiotic is a combination of probiotics and prebiotics both, and metabiotics is actually using the bacterial waste. So you allow the bacteria to eat their food to produce metabolites, and you eat that waste product because it is good for your own body. And this waste product, though it is called as waste product from the bacteria's point of view, they contain short chain fatty acids, which are actually neurostimulants. Okay, they also contain vitamins, many other important signaling molecules. So, in case of genitourinary tract, the normal microflora generally consists of the M. smegmatis, which is basically found in both males and females. The Gardnerella vaginalis, which is generally found in case of penile urethra. In case of vagina, the microflora changes as per the age. Like in case of before the puberty, there is no glycogen which is present in the vagina, and therefore it has a high pH. Okay, after the puberty, the glycogen is used by the microorganisms as a food, and therefore they ferment it to produce lactic acid. Like especially the bacteria is called as Lactobacillus acidophilus, and this is how it appears. You can see at the bottom. This bacteria basically produces lactic acid from the fermentation of the glycogen, and therefore making the pH acidic in the vagina. And after the menopause, when the menopause, the pH of the vagina again increases to higher level, just like pre-puberty level, and therefore the pH becomes alkaline, and it leads to the glycogen-free environment. So, what are the pros and cons of the microflora if you compare the overall microflora of the body? Basically, there can be beneficial effects, like they can be exclusion effect. So exclusion effect is basically excluding the pathogenic bacteria and other organisms. So basically, how does normal microflora does that? Because normal bacteria, they colonize your body parts and they make a community. So this community produces many metabolites which doesn't allow the other bacteria to colonize. And these bacteria generally are the pathogenic organisms. Okay. In case of immunostimulation, this bacteria produces, as I said, so many metabolites in your gut especially, and this gut metabolites, they affect immune system of your body, and they also affect your behavior. Okay, and many of the gut microflora also produces essential nutrients such as vitamins and other cofactors, etc., etc. However, they also have a harmful effect. Okay, and this harmful effect is that because the microorganisms, they reside in your body, this microflora reside in your body for a long period of time, they can also mimic. So this microflora, this bacteria, they can also mimic the normal tissue antigens which are present in your body. And by doing that, they can make the cross reactivity happen. So why I'm telling you is that, so basically, let's assume that a bacteria in your gut has an antigen. Antigens are proteins. So basically, the bacteria has an antigen or a protein which is basically mimicking the protein of your other body part. So if the antibodies against that protein comes to your body, they can hamper both the gut microflora as well as that other normal tissue. So there is a cross reactivity. Also, they also can cause the opportunistic infection. I told you that the true pathogen and the alternative facultative pathogen. Okay, so this normal flora can also cause opportunistic infections if they're given a chance, and they can also produce carcinogens in a long period of time. Okay, and the areas of the body with flora we had already discussed. So we have discussed about the microflora of the human body. Now we'll also discuss about how this microflora play a very essential role in making the many things possible. So if you talk about making cheese and yogurt from the milk, all of you know that the milk is basically kept for the fermentation, and this fermentation is done by lactic acid bacteria, and this lactic acid bacteria then what makes the curd and the cheese from the milk. Also, if you dough your if you knead your dough, आटा जिसे गूंदना कहते हैं, if you knead your dough and you keep it for some time outside of the out in the room temperature, you will slowly see that the dough it basically the फूलता है dough. So why it is so? It is because of the yeast. Okay, and therefore, and this yeast basically makes it soft. It also degrades some gluten. Okay, so to the carbohydrates. So generally now what is done is that generally the baker's yeast, it is called as baker's yeast, that is the Saccharomyces cerevisiae, and this baker's yeast is added from outside to the dough to make it very soft and fluffy. Okay, and along with that, the fermented food products like that you eat, like the dosa batter, the idli batter, okay, all all of that is made with the help of microorganisms, and also the brewing industries, which basically makes alcohol every day and every night. So like beer, all these things, beer, wine, all of these are made with the help of microorganisms. Okay, so now you can understand how microorganisms are important from every single aspect. Okay, I thank all of you, and you should respect your microbiome, your body's microbiome. You should not disturb them. Okay. Now, I also thank the GD Goenka University for giving me an opportunity to ignite younger minds toward the exciting field of microbiology. And I primarily thank the School of Basic and Applied Sciences, the Dean, Professor R.K. Thakur. Uh, Professor R.K. Thakur, he has just received an award for his significant contribution in the field of nanotechnology from the hands of Krishnan Reddy, who is the Minister of State and Home Affairs, Government of India. So let's talk about the faculties of the School of Basic and Applied Sciences. From the physics, we have 6 faculties, and they are from eminent institutions of India and abroad, basically, and these are some of their research interests. If you talk about chemistry department, we have 8 faculties, and all of them again are from eminent institutions of India and abroad. Most of them are from IITs, and they also work on different areas and conduct their research. The mathematics consists of so many faculties from different institutions of India, mainly IITs and NITs, and they also work on different research interests. In case of biological sciences, we 3 of us are there. We work on so many different aspects of biological sciences and conduct our own research. In case of, we also have forensic science department, which consists of 5 faculties, and these faculties, they play a very essential role in conducting research at various levels. The research at SoBAS consists of so we have around 30 Ph.D. scholars which are registered in various departments of the school, and they work in biological sciences, physical sciences, chemical sciences, mathematics, environmental sciences, etc., etc. 3 of the projects have been sanctioned by the government of India. 2 of the projects have been sanctioned by the government of India, and 1 project have been sanctioned by the consultancy project. So we also have a state-of-the-art lab facilities for each department, where the students, they conduct their experiments and enjoy the hands-on training experience. The programs, the UG programs in SoBAS are there in chemistry, physics, mathematics, forensic sciences, microbiology, biochemistry, and biotechnology. And also we have BSc pass courses, which consist of so many combinations. In case of the postgraduate and doctoral program, we have MSc in chemistry, physics, mathematics, forensic sciences, microbiology, biochemistry, biotechnology, data science, geology, wildlife, forestry, biophysics, and environmental science. Along with that, we also offer PhD programs in so many different domains. The PG diploma courses are also offered by the SoBAS, which consist of diploma in criminology, cosmetics, data science, environment, health, and safety. We also follow practices which consist of mentor-mentee relationship. So each student has a particular mentor, and they basically reach their mentor if they have a problem. If the mentor cannot solve the problem, then they will reach to the HOD, and if the HOD can't solve the problem, then they go to the dean. And at the end, the dean solves the problem. We also have a SIM login system where individual student can visualize their attendance, their marks in a centralized manner, in an online centralized manner. We also carry out so many experiential learning activities, which basically consist of educational trips to industries and laboratories, conferences, and the faculty development programs, the student development programs, workshops, symposiums, etc., etc. We also invite lots of distinguished and expert guests to give a lecture in our school, and we also take our students to the long and short-term trainings at the industries and the institutions. So you can see a talk by the distinguished scientists from NASA, Dr. Kumar Krishan, and also by Professor P.B. Sharma, who has been the Vice Chancellor of 2 different universities, the founder and Vice Chancellor of 2 different universities, and currently is the Vice Chancellor of the Amit University, Manas. In terms of experiential learning program, we also invited Dr. D.K. Aswal, who is the director of NPL, that is the National Physical Laboratory. We also invited Professor Ram Kumar, who is the Dean in Central University of South Bihar, and also Dr. N.B. Bardhan, who is the youngest director of the CBI, CFSA, Delhi. In the case of student development program, we also celebrated Charles Darwin's birthday under the event named Creation. Creation 2020. And we also invited speakers from the Department of Biotechnology to deliver their lecture on bioentrepreneurship and the use of it as a career opportunity. Uh, Also, we have taken our students to industrial visits to NPL, IIT Delhi, and IUSC. Also, we have taken the students to Anveshana, which is basically a science and engineering fair held at National Bal Bhavan, New Delhi. And few of our students have also won an award in this event. And some of our students have also visited the AIMS, basically the Forensic Science Department. We also have been a part of National Online Conference, which has been carried out by the ESDA, Delhi. And also we conducted a conference, international conference, basically on sustainable cities and communities in Feb 2020. Some of our faculties have also visited several schools to basic as a part of school awareness session and as a school kind programs. We under the school connect program, the students from various schools from Delhi, Gurgaon, Haryana, Rajasthan, UP. Okay, they have visited our school in the January 2020 under under the DST Inspire camp, which is basically funded by the Department of Science and Technology, Government of India. And this camp basically provides a great opportunity for the younger minds to interact with the renowned scientists of national and international repute during this 5 days of science camp. So in this camp, we also invited so many different scientists from reputed scientists from all over the India and abroad, and they give a very good they give very good lectures, which basically motivated these younger minds towards this science education. We also have training and placement cell, which basically has tie up with so many companies, and these are some of the placement which are done in the last year. So for more information, you can visit our school. We have a Facebook homepage also, and we also have an Instagram homepage. Thank you.
Well, thank you, Dr. Kamble, for such a nice, fruitful, and informative talk. I'm sure all our students and attendees have improved their understanding. And moreover, now we open the house for the questions. So, Dr. Renu Chaudhary.
Yes, sir.
So, could you please ask the question on behalf of attendees?
Yes.
I think there are a lot of questions.
Yes, sir. Sir, first question is from Ankit Sen. He is asking, our environment is consist of millions of viruses. Still, our immune system can protect against them. But I want to know that against which kind of viruses our immune system fail to protect us and why.
See, viruses are 10 times smaller than the bacteria. Okay, they are very small than the bacteria. So when your immune system produces antibodies against the viruses, basically these antibodies, what they do is that they bind to the viruses and basically prevent its binding with the host surfaces. And that is how they prevent the entry of the virus into the host cell. So the viruses which can escape from this immune system are basically the viruses which can mutate very quickly. And the best example of is the influenza virus. Every year in some part of the world there is an influenza outbreak. Okay, and this is not the new strain, this is the same strain which basically mutates and becomes a new strain. Okay, so influenza is the best example of such a virus which can escape from the host immune system. Okay, and can cause the disease in multiple individuals.
Okay, sir.
Yeah.
He has again asked, what is the mechanism of immune system to act against such kind of viruses?
So there are two mechanisms. First is that viruses are obligate intracellular parasites. That is, viruses cannot survive outside of the host cell. Okay. So viruses, when they infect the cells, this infected host cells, they become abnormal. Okay, and this abnormal host cells, then they can be killed by the your own immune cells. And also, as I said, the antibodies which are produced by the immune cells against these viruses, they can help to minimize the binding of the virus particles with the host cells.
Ankit Sen, are you here in this meeting? You may turn on your mic. Mr. Mr. Ankit Sen.
Ankit saying you want to understand the mechanism. See, the mechanism can be classified into so many types. Okay, so if you know about your immune system has three different what you can say types. First is the first line of defense, second line of defense, and third line of defense. So first line of defense is the physical and physiological barriers which prevents the infections from the outside microorganisms. Okay, and many of your immune cells and also your body, it produces many of the antimicrobial and antiviral defense antiviral peptides which can basically kill the invading pathogens. Also a very essential mechanism of your body to kill the viruses is basically fever. So I think you know that interferons are the cytokines which are produced during the viral infection, and they raise the body temperature. It is because if you raise the body temperature, the viruses can't multiply. Okay, and thereby their infectivity is limited. And also along with that, the third line of defense, which consists of your humoral immune system as well as the cell-mediated immune system, it consists of antibodies and the immune cells. So the antibodies can prevent the viral binding to the host cells, and the immune cells can kill the viral infected cells. So this is the overall mechanism of viral neutralization.
Yeah, Dr. Singh.
Yes.
Moving on, other question.
If you have more question, Ankit, you can call me on my number. Okay.
Renu ma'am, I think there has been a discussion between Kanchan and Mervin. They have, so I believe we can just ask them if there is anything else they want to ask Shashank sir. Kanchan, is there anything you would want to ask?
She has written since the newborn is getting exposed.
This was actually answer to Ankit's question, I believe.
So there's a question from Sangamitra.
Yeah, maybe that is something that can be answered. And if Kanchan and Ankit, you still have some doubts, you can ask Shashank sir individually through call or email.
Sonika ma'am, I think Kanchan was giving the answer to the queries open.
Yeah,
exactly. That's right.
Yeah.
So Sangamitra's question is that can we cultivate all the body macroflora in a lab for studying their growth phase of further studies? You cannot cultivate all the body macroflora. I tell you why. Because if you talk about gut only, okay, the gut has partially anaerobic parts also, and the part which are almost anaerobic, fully anaerobic. And it's very difficult to make such anaerobic conditions outside in the laboratory conditions. Okay, in fact, I would tell you that there are some bacteria which were known to be present in the human intestine, and they were known to like we talk about the inducing the host immune system, right? So there's a bacteria called as SFB, segmented pharmatic bacteria, and that bacterium we have been able to cultivate it after 70 years of its discovery. So now you can understand how difficult it to grow it in the laboratory. So you can't cultivate all the bacteria. So that means if you are right, Ankit.
Yeah, any more question?
No question, sir.
I think I saw so many questions.
If you have more questions, you can reach me on my number also.
Dr. Naresh Sharma ji.
Hello. Ji sir.
Ji, Sharma ji, quick question. Is there any question from the student?
No sir. All are answered, sir.
So quickly. I
All
thought
are
today
answered,
the question answer session will go for 1 hour. All answered quite smartly. Thank you. So I think then we should thank the organizer and the speaker. So thank you, Dr. Shashank Kamble Sahab, for such a informative talk. I think it is a general aspects and helpful for almost everyone. So thank you once again. And also I extend my thanks to the coordinator, Dr. Renu Chaudhary, and her team, Dr. Naresh Sharma, Dr. Sonika Sethi, and Dr. Kamble, and all other faculty members of SoBAS. And also I extend my thanks to all the learned people who have joined from outside and our students. So thank you so much. I think next session is on, do we have an advertisement? No.
Sir,
But I can remember.
yes, sir. Sir, we have next session, sixth session of SDP series on 26th August 2020 on Fun with Physics. We
Okay.
have Dr. Shashank Gupta and Dr. Sunita Negi, Assistant Professor, Associate Professor SoBAS.
Good. Okay. So it is on a fun with the physics, right?
Fun with Physics. Yes, sir.
Okay, great. So it is a day after tomorrow then, 26, right?
Yes.
Yeah.
उल्टी आ रही है तो
26
उसके बाद जैसे वेक्टर जाता है अंदर मुझे शो होमवर्क करने का मन नहीं होता क्या मुझे भी लग रही है इसमें क्या काम कर रहे हैं बैठे फिर भी
Hello. Ji. So, well, so next session is the day after tomorrow. So I may request, yeah, here we have.
Yes, sir.
Yeah, good. Yeah. So next session is on day after tomorrow, the SDP.
Excuse me, sir. This is about eminent scientist series.
Right.
For SDP, Renu ma'am just said that's on 26th. So,
Okay, 26 SDP,
Yes.
and this is the eminent scientist webinar series on 28th. So on Friday. So here both of our speakers are from IISc Bangalore, and they are a physicist and electrical engineer come physicist, and both they obtained a PhD in America, and they were in the United States at Harvard University. Harvard, you know, quite very famous, I think one of the most famous, for more than 5, 6, 7 years. And the other was also at Caltech, California Institute of Technology, Pasadena at United States. So both agreed on our request to give a talk on nanotechnology training and research at SENSE. SENSE stands for Center of Nanoscience and Engineering. It was actually inaugurated by our Prime Minister, Mr. Narendra Modi, in 2015, I suppose, I guess, 14 or 15. After turning the Prime Minister, he has laid the foundation stone, and the SENSE houses more than, I think, 20 or 25 very senior professors, and they are working on a nanotechnology, providing a research, training and research to the people, not only from this country, but also from across the world. So both our talk, the first one is day after tomorrow is a SDP, Physics with the Fun, and afterward on a Friday, we have this webinar series. So I may kindly request all the LoveNet members, all faculties, students to join this talk and also disseminate information among the peers, among the colleagues, because these are really very important information. You know, most of the people, one survey says that most of the people, more than 90% people, they did not get, say, correct height in their career and profession just because of the information. They won't get any information, and due to the lack of information, they are unable to touch the height where in principle they should be. So all our motive is to provide information as much as possible, whether it is a subjective information or related to the career or placement or whatsoever. So once again, thank you so much indeed. Thank you, Dr. Kamble. Thank you, Dr. Renu Chaudhary. Thank you, Dr. Sethi, Dr. Naresh Sharmaji, and all the faculty members of SoBAS and external members who have attended this talk. And hope to see you soon. I wish you a nice and wonderful afternoon, nice and wonderful evening. Stay healthy, stay safe, and stay at home. Thank you.
Thank you.
Take care. Namaskar.
Thank you, sir.
Thank you, sir.